had it with these bloody Americans. I've had it with it. I didn't want to come here to this land. King George's order to me, go take back the colonies for the motherland of England. These damn Americans, bloody Americans. They come and they shoot us and they leave, and then they come and they shoot us and they leave. Bloody Americans, and listen to their flutes off in the distance. I've heard more flutes than I ever want to hear again. I'd rather hear a good sea ballad or something like that. I've been outside of Baltimore for more than a fortnight in an encampment. But every night there's raids. We're out of food, I had to eat my trousers. It drives me mad. All I want to do is go back to the lovely rain of the British Isles. We don't like the rain here very much. But anyway, I don't know what to say. Except for I cannot hear another American song. I think what I'll do is I'll teach psychology instead. And maybe we can see if the musicians will play a good ballad. Ah, never mind. All right, hello everyone. We are going to talk about psychology. We're going to talk about, there it goes. We're going to talk about neuroplasticity. That's right. Neuroplasticity, what's that, Mr. Stewart? I'm not Mr. Stewart to you, bloody Americans. I'm the feared commander of something, but I'm feared anyway. So you have to say, what's neuroplasticity, feared commander of something, but feared anyway? I'm going to tell you. This is actually, okay, seriously, this is actually really, really important. This neuroplasticity and its implications. And so let me explain some of the things here to you. What is neuroplasticity? It's very important. It's one of these things you have to know. You have to know. Have to know. Neuroplasticity is the ability for the brain to adapt its neurons to the environment. And this can be both good and it can be bad. If you are in a bad, abusive environment, then neurons and part of your brain, they basically die. If you're in a very sort of thriving and helpful environment, then you have more neural growth in your brain. It can also change in older in life. If you do a skill, if you like play music, an instrument or something like this, and you get really, really good at it, you can actually see a difference in the brain. That your brain actually changes, it adapts to the sort of the situation you're in. There's a number of experiments we'll look at, one with juggling, one with taxi drivers. There's a number of them that talk about this. And neuroplasticity is absolutely, absolutely critical for the human brain and human personality. And so we're going to talk about this substantially. I'm going to do a basic lesson on this today. But anyway, neuroplasticity is this. And its implications, environmental factors that influence the brain's physical structures, okay? Environmental factors that influence the brain's physical structures, and that's just what I was saying, that in a positive environment, a good environment, you have a lot of, good, so you get more brain growth. In a poor environment, you get less brain growth. Kids who are neglected, their brains don't grow. Kids who are, taken care of properly and given attention and given love and so on, their brains grow. They become better adults. And yes, so we'll talk about this more. This has a lot of implications for a lot of mental health issues. You see that a lot of the people who are on the streets have mental health issues, and a lot of those people also have very poor home lives. And there's a very strong connection. It's correlation. It's a very strong connection, though. You could almost say it's causal, but not really. But anyway, so happens to do a different couple things. Dendritic branching, I'll talk about that in a minute. Remember what the dendrites are? The dendrites are those little things. We're having some diagrams, some pictures in a second, those little things at the end of your neurons. Okay, that's where the actual sort of stuff is taking place, the neural transmissions are taking place, and the neurotransmitters go out of there into the next one. Well, if there are a few of them, there's going to be fewer neural transmissions and so subsequently less activity. And if there's less activity in, for instance, the parts of your brain that have to deal with empathy or have to deal with, intelligence, I guess, if those are less, then, well, everything's going to be less. And if they're more, it's going to be more. We'll talk about that more. Enhanced environment provides stimulation like social interaction and learning. Social interactions and learning provide stimulation for your brain, and it makes your brain grow. If you want to have smart kids when you end up having kids, which you do, I'm sure, want to have smart kids, read to them, get them to do things, don't stick them in front of the tablet, get them to, have lots of experiences and be part of the family and that type of stuff, and that helps a lot. Okay? Experiences processed by the brain resulting in an increased number of synapses. This is the thing. You have more synapses, and if you have more synapses, you have more activity. And if you have more activity in those parts of the brain which are, help you succeed, then you are going to have more activity and you will be a more intelligent person. You will be more intelligent, simply, and more empathetic and more, you'll just have more skills than somebody who does not have that. And they have shown this in animal research. You can't do it with humans, obviously, stick a kid in a box, although it's happened. But unfortunately, there's a lot of neglected children or abused children. Those are not necessarily, well, neglect is abuse, but abuse is more than neglect. So, but neglect, they talk about neglect and like physical abuse as two different things. But they have kind of the same sort of, I guess you could say, result in the development of a child's brain. Okay? But anyway, so experiences processed by the brain. Yeah, I just read that. Animal research, and then case studies on neglected children. So we're going to look at a very famous animal research study. Then we're going to tie that in. I'll see how long that takes. Then we're going to tie that into another study. I may do it in another lesson. We'll just see how long it takes for me to get there. And so this is really quite fascinating. It's super important, this stuff. This is like the central core idea of stuff. It's like very, very important for neuroscience today. And if you go on to study psychology, which I hope some of you do, if you go on to study psychology and think it's a fascinating thing you want to learn more about, you will be doing this type of stuff, neuroplasticity. It's very leading edge. It's very sort of like, you're not doing neuroplasticity, but you'll be studying that sort of neural growth and things like that. It's maybe not at a bachelor's level, but when you get higher up and research, there's all kinds of work to do with this. Tons of it. And it's fascinating, absolutely. Anyway. Dendritic branching, okay? To the left, I'm going to put this little diagram up there above the tree. Above the tree it goes. And you see to the left, you see an enriched neuron. And to the right, somewhere in here, you'll see an impoverished neuron. Okay? Now, you also see a very excellent, on the left, and on the right, very excellent ideas for Halloween costumes. You can go to a Halloween party with a friend, and one of you can be an enriched neuron, and one of you can be an impoverished neuron. All you need to do is get a bunch of cigar cleaners and make a funky hat. I'm an impoverished neuron. But what it is so what you see there is a kind of just a simple written, drawn representation of what a neuron will look like that has a whole bunch of things on the end. And those whole bunch of things, those are actually brain mass. And remember how small your neurons are. You have billions and billions of them in your head. So you have, if you, multiply that brain mass by billions and billions, and if you multiply the other brain mass by the impoverished brain mass by billions and billions, well, you can see the idea that you would have more brain mass in your brain than somebody who has a whole bunch of impoverished neurons in their brain. And so all of these centers of your brain that function and help you do things will have less. And you will have less, a person will have less intelligence, less ability to interact socially. Those are really the things which get damaged. It's not going to damage your, it damages everything, never mind. But yeah, those are things which are primarily damaged. And a lot of people with, I guess you could say, what's the disorder? Antisocial personality disorder. They think they think that a lot of people with antisocial personality disorder probably have disruptions because they can't deal. I mean, it's some social personality disorder is somebody who like fights all the time or causes problems all the time and all kinds of stuff. And there's some research talking about the relationship between antisocial personality disorder and neglected children. Okay, so there's a connection. But anything, I'm not getting to that right now. But so what I will do is, so what you want is you want the enriched neuron and you don't want the impoverished neuron. And the way that you get that is by social interaction, by caring environments, by proper nutrition as well. But you can have proper nutrition and being neglected and you'll still end up like that, impoverished one. And so let's go on there. Famous study, know this one, know it, just know it. It's really, really good one. Most of the studies I talk about are ones you really need to know, if you haven't gathered yet. How do I look in the do? I look good in the do. It's a little bit, bloody Americans have messed up my hair. But besides that, so here is the name of the study. The people who did it were Rosenweig, Bennett, and Diamond. 1972. It's an old study, but still a goodie. Oldie but a goodie. All right? The role of the environment. The role of environmental stimulation on brain plasticity. So here's what they did. They took rats, right? And they placed rats in an enriched or deprived environment. Here's what they did. Here's what they did. The enriched condition, 10 or 12 rats per cage with things to explore and play with. So they put them in a cage. They're cage rats, but they don't know anything else. So they put them in there. They get to play with the other little cage rats. And they get little balls and little fucking funky running things and little tubes to crawl through. And you can imagine a happy little lab rat habitat. Oh, look what I did there. A lab rat habitat. That is gonna launch my career in rap. The lab rat habitat. So it's as happy as a lab rat could be. Boom. They don't know anything else. And then in the impoverished condition, they just stuck a rat in a cage. And this is when they were babies, young, okay? When they're just babies, just right after they're weaned. Or they can even feed them themselves, but they're very young. Rats don't take long to get to maturity. So they stuck just in a cage. No toys for you, little rat. You did something bad in a past life. In a cage. You had food, had what it's needed. It doesn't get to play with anyone. It doesn't get any toys. Just sit in a cage. Right? And so the other rats are doing their little rat things, and that one just sits in the cage. Okay, so what happened? What did they do? So that's the idea. And then they killed them. They did. They gassed them with carbon dioxide. That's how they kill things. But anyway, so what they did is the brains of the EC rats, enriched condition rats, they had increased thickness and higher cortex weight. Meaning the cortex of their brain, it was thicker and heavier. There was more brain. The enriched condition rats had developed more acetylcholine receptors. Acetylcholine, remember, acetylcholine is what is needed to learn. Your hippocampus has acetylcholine receptors. And so when you're learning through the hippocampus, when you're learning, your acetylcholine is firing, the acetylcholine receptors are firing, and it's encoding short-term memory to long-term memory. And there's other things going on with acetylcholine, but these rats had far fewer, the ones in the poor condition had far fewer acetylcholine receptors. Acetylcholine receptors are associated with learning. They were stupid rats. The other rats were happy rats, smart rats, smart as a rat can be. But they were fine. They were just normal rats. But the ones who were put in the impoverished condition were stupid rats. They had, their brains were not good. And so some other things are kind of interesting. Since they do it here, no. Another thing that's interesting about it is the behavior of the rats. They took some of the rats that had been, raised in the cage like that by themselves, and they tried to put them into the community of rats to see if they could turn into normal rats. Didn't work. The rats they fought all the time, or they sat in the corner and cowered all the time. They were not able to be socialized into being normal rats. Rats very apparently have also a critical period of development. I've talked about critical periods of development many times in class. And we have them, definitely, absolutely. And rats apparently have them too, because these rats who were damaged rats, they could not be normal again. Their brain was broken. So, okay. And so it's very, very interesting. They did a bunch of different conditions with it. Some rats later in different trials, what they would do is they would take them out of the impoverished cage and they would stick them in the normal cage, like for an hour a day or something like that. Those rats turned out fine. It was only if that severe neglect, that extreme neglect, and lack of social interaction and lack of learning and that kind of stuff, that was really what was damaging. Even if it was just for a little bit each day, the rats were okay, a couple hours a day. It wasn't just like 1 minute or anything like that, but it was, a couple hours a day. Evaluation of the study. It was a well-controlled lab study, okay, sure, in a lab, good variables and all that kind of stuff. But anyway, there's animal models or problems for human models. Unfortunately, we have enough evidence to show that this works for humans too. It's probably why we're still talking about this one nearly 50 years later. But anyway, so animal, so it's you can't necessarily transfer the animal study results to humans. We're not rats. Our brains are far more complicated than rats. We're not rats. We don't behave like rats. We learn a little bit like rats do through trial and error, but everything kind of does. But so you see what I mean. Except for cows, I don't think they learn. I guess they do. I don't know, but they don't look like they learn too quickly. Rats learn pretty quickly. So, ethical issues, is it justified? That's a good question. Is it justified to take these rats and just do this experiment on them and basically torture and neglect these rats and then kill them and look at their brains? I don't know. Could there be warranted reasons for understanding models of human neglect? Yes, there is. And so, as long as the animals aren't undue suffering, and you could say that the rats in those cages probably were, but still, was the knowledge worth getting? Yeah, probably. It's an ethical dilemma there, and you can't really hands down say, hey, no, this is absolutely unethical. You can't really hands down say it's totally ethical either. But it's an accepted study. They are just rats after all, which is easy for me to say when I'm not one of them, but they are just rats after all, and we do use animals for human benefit. None of the medicine we have was not tested on animals. All of it was. And, you kill a lot of animals before you get a medicine that works for humans, and that's just kind of something that happens. Whatever the case. Okay? Okay, so let's get that. Now what I'm going to do is I'm going to talk about, I guess I'll talk about this one. How long are we going? Yeah, I'll have enough time to talk about this one. This is actually really a good one. I'm just trying to see something. You know what? I'm going to talk about that one in class. This one that I just went over through here. That's a really good one, and I actually want to talk about it in class. Yeah, I think I'm going to do that. So I'm going to just go to this one, and this one's kind of interesting, and I'll just do this, and then and this is also about neuroplasticity, and I'll do this, and it'll be good for the lesson. And then when I do this lesson in class, I'll talk about the Perry one. It's a really good one. I kind of want to do talking and kind of show you the anyway. And so anyway, so the aim of this, and now this is kind of an interesting study. Dragonsky, 2004. It's an excellent study to illustrate neuroplasticity. You very commonly will get a question about neuroplasticity. It's a very, very common topic to get in the IB exams. They talk about it all the time, either in the short questions or the long questions. Very commonly, there's one in there. So you got to know it. You got to understand what's going on here. So here's some studies. And we're going to study, several of them, so you have enough material to answer really well if you get a longer question on it. But determine whether functional and structural brain changes could be detected in the human brain as a result of learning new motor skill. So they said, okay, you're going to learn something, a new motor skill. It was juggling. But you could pretty much, I think, translate this to any new motor skill, like playing a piano or an instrument or something like that, or getting really good at some sort of a sport or something like that. It makes sense that all those come with some sort of neuroplasticity and changing of your brain to allow you to do things, because you actually get better at them. and something's happening. There's a reason you're getting better at that. There's something happening in your brain. Call it muscle memory, but your muscles don't have memory. It's your brain, and your brain learns how to do it. But anyway, so 21 females and 3 males were split into 2 groups. One group had to spend 3 months learning to juggle for a minimum of 60 seconds. Juggling. Can you juggle? I can juggle. 3 balls. No problem. I can juggle for 60 seconds. My brain is going. Okay. And then spend 3 months not practicing juggling. Okay? So, some people were told to juggle and some weren't. And so basically it's simple. There's really not much more to that study than that. But here's the interesting thing. MRI scans showed no structural differences in groups' brains before juggling. So they stuck them in an MRI. Like, gosh, whoops, I'm talking too fast. I'm thinking too fast, and my mouth isn't catching up. They put them in an MRI and did MRI scans on their brains before they started the study. That is what I am trying to say. But afterwards, after the study, there was an increase in the volume of 2 regions of the juggler's brains associated with the retention of visually detected movement, information, and learning. Right? So, in certain parts of the brain, there was an increase in mass after doing the juggling thing. So if you want to increase your brain mass, juggle or do something. It's probably, I am 100% sure, although have never seen the thing, so I can't be 100% sure, but you know when you play video games? I play video games. You know when you first get the control of your video game? And if you don't play video games, you won't know what I mean. But if you do, you totally will. Or you use a keyboard, whatever, it doesn't matter. And you're playing, and you're like, it even happens a little bit when you start a new game and the controls are a little bit different. Your guys jump is when it's supposed to slide and stuff like that. But you play. But after not very long, especially if you play it a lot, you're just like, you don't even have to look at it. You can be sitting pitch black and it doesn't even matter. You just totally know what's going on. And so that's something going on in your brain. Absolutely. I'm 100% convinced. It would be super interesting to do a study on that action, is to get gamers and non-gamers and study their brains before and after and whatever the case is. I'm sure you would find somewhere in the brain that your brain mass is going to be more in a person who games. It doesn't mean you're smarter, it just means that in those areas of the brain associated with that activity, I'm sure your brain mass would be more. It's the same sort of idea as what they do here. So there was an increase in there, and the interesting thing is that increase went away after 3 months of not juggling. So the conclusion, practicing watching balls move and learning to move in response has strengthened the neural connections of the brain areas responsible for the activity. Okay? So, see what I mean? That's what it is. And I'm sure it's the same way with, that visual hand-eye coordination sort of thing going on with playing the video game controller or whatnot, or anything that you do. And so, okay? The study demonstrates the effect of enrichment on the brain plasticity. This is basically a, let's see what the next one is. How long have I gone on this one? I think I'll do one more study on this one after this. This is just something for you guys to read. This is a connection to the study question slide. And what that means is that it's kind of a tie between how you would answer the question. Like if you were to get a question, evaluate 3 studies on, or not evaluate, illustrate, or whatever. Evaluate 3 studies that illustrate the plasticity on the human brain, or something like that. If that was a question, I could find you a real question. I'd have to look it up, and I'll show you those later. There are questions about that. This is kind of how you would make the connection between the study and the question. It's a slide for that. It's really a slide for you to use, and not for me to go over in a PowerPoint. It's more of a sort of, me giving you some notes type slide. So just read that. I'll do one more study on this, and then I'll call it good for this, because I don't want to talk too long on those things. But this is also another one. The Maguire taxi driver study in London. Very interesting study, very good one to use. Remember this one too, remember all of these. Aim to investigate whether changes, plasticity, neuroplasticity, could be detected in the brains of London taxi drivers and further investigate the functions of hippocampus and spatial memory. So I don't know if they still do it when there's Uber and stuff, but traditionally, before there was GPS, the London taxi guys had to take these complicated tests and learn the entire map of London. And London is like the roads are crazy. I mean, in our cities in the US, they built them on a grid pretty much because we built many of them after there were already cars, so we built them in a way. But in the old European cities, they were built around the church, and there was no cars or anything. They were built around the church, and the cities evolved kind of concentric circles around the church. Usually there's a church right in the center, and it goes out like that. So they built all these different streets and roads and aisles or alleys and walkways and stuff like that. There's a zillion of them all over London. And if you ever go to London and just walk around in an area, it's actually kind of cool. There's so many little streets, especially central London, the old parts of London, not necessarily the suburbs that are built on a grid. Anyway, so the taxi drivers need to learn all this stuff, okay? And so they probably have GPS now, I don't know. But anyway, so if there were any differences in the brains of those who rely heavily on spatial navigation skills in their working life, these differences would be found in the hippocampus, because the hippocampus is responsible for transferring short-term memory to long-term memory. So if it's like getting a lot of activity, it should be bigger. Participants, 16 healthy right-handed male licensed taxi drivers, 44 years of age. All participants licensed London taxi drivers for more than 1.5 years. He had a control group, somebody who was not taxi drivers at all. No women or left-handed people. All right, that went pretty quick, but that's the idea. Those participants there. I can put it back up there real quick. You take a look at it. So remember, you have it in front of you, or you can have it in front of you very, very easily. Okay. So what they did, they put them in an MRI. They were performed by measuring just the taxi drivers against non-taxi drivers. So they took people who were already taxi drivers for at least 1.5 years. Then they just did a comparative study with people who were not taxi drivers. And they wanted to see if there was a difference in the hippocampus volume. And so the MRI scans were performed by measuring tissues. Measures of density of gray matter in the brain, VDM, provides a 3-dimensional measurement of the volume. They did pixel counting, measured the volume of the hippocampus. Okay, so what did they find? They found exactly what they were thinking they would find. They found that taxi drivers had an increase in gray matter in the left and right hippocampus. The increase was limited to the posterior. The rear hippocampus is way inside your brain. Okay? But the hippocampus was bigger in these taxi drivers, and it was in the rear part of the hippocampus. If you have left, right, front, back, it would be in the posterior, the back side of the hippocampus. They had a greater volume in the anterior, okay, and in the anterior. The increase was limited in the posterior, but it was still there, and they had a greater volume in the anterior. I mixed that up, I read it wrong. No difference in the overall volume of the hippocampus compared to the control group, but it was different in comparing the regions. So what happened is the hippocampus itself was not necessarily bigger, but different parts of the hippocampus were different. And so that's the interesting part, is that the brain changed. The hippocampus, the structure, the actual physical structure of the hippocampus changes when you're learning something again and again and again and again and again and again and again. You get better at it. And I'm sure as you learn more, as you get older, your brain probably, when you go to college and you learn all these things and you know all this stuff and you're remembering all these facts and so on and so forth, your brain probably just adapts and gets the sort of, plastic changes that adapt to what you're doing. Okay. All right. So evaluation of the study, strengths, the scan data. It's really good data. It's really, really good data. So, it's like you can't argue with the data itself. It's done on a machine. It suggests that the hippocampus increases with size as a result of navigational experience. It's well controlled. But it's well controlled, the experiment, or the thing itself. It's not really an experiment, it's data collection. So an experiment would have it would have a independent variable, which is something that changes. So they're just looking at people. So this means that it's kind of a pseudo experiment, a quasi experiment. And so the weakness of this is these people, they didn't have control of the people. They took 16 taxi drivers, 16 other people, and they take a look, and sure, it shows a difference, but we don't really know what the life of these 16 taxi drivers or these 16 other people were like. beforehand, they're only looking at a particular element in time. So maybe the people with bigger hippocampuses want to be taxi drivers. Maybe the taxi driver doesn't cause it, they just have it. Who knows? Probably not, but you see what I mean. It's not really well controlled with participants. And so that leads a weakness to the claims there. It was only right-handed men. Okay, which it probably no real big difference, but whatever the case is. They just chose right-handed people. Okay. And so a good study, well done, showed what they wanted to show, but the thing that it's not really an experiment, it's more of a comparative of people who haven't controlled their lives. That takes a little bit away from the sort of validity of the claims, but it doesn't mean it's wrong. It's still a well done study. All right, with that, I've got to go back and fight these bloody Americans. Bloody Americans made me eat my own trousers. See if I find some better music now. Hopefully. There, maybe it's this.
An American frigate, a frigate of fame.
That's better.
With guns mounted
Off I go.
Richard by name.
And remember, you will never be allowed to leave England and the British Isles. King George shall have you for eternity.
We had not sailed long before we did espy a large 40